Every episode so far has treated molecules as flat 2D graphs. But a drug actually works by physically fitting into a pocket on its target protein, like a key in a lock. Today we go three-dimensional. We download the real EGFR protein structure, and we dock a drug into its binding site, predicting both how it sits and how well it fits, using open-source tools and a few lines of code. Docking has three ingredients. A protein structure, which we get from the protein data bank, real coordinates from X-ray crystallography. A pocket, here the ATP site of the EGFR kinase, where its inhibitors bind. And a score, a number in kilocalories per mole that estimates how tightly a molecule fits. The engine tries many poses and reports the best. More negative means tighter binding. First, the structure. We pull PDB entry 1M17, the EGFR kinase domain crystallized with the drug erlotinib actually bound inside it. We split it in two, the protein atoms become our receptor, and the co-crystallized drug becomes a reference. Then open Babel adds hydrogens and charges and writes the PDBQT files the docking engine needs. Real structure, minimal fuss. Now we dock. We hand smina the receptor and our query molecule, gefitinib, and use the crystal ligand to auto-size the search box, so we're docking into the true ATP site, not guessing coordinates. It searches poses and scores each one. The best comes back at -8.8 kilocalories per mole, a strong predicted binder, exactly what you'd hope for a real EGFR drug. But the number alone isn't enough, the pose has to make physical sense. So here's the payoff. In yellow, our docked gefitinib. In blue, the crystal erlotinib that was actually in the structure. Look how they overlap, the same quinazoline core threads into the same pocket, the halogenated ring points into the same back cleft. Docking recovered the real binding mode on its own. That agreement is what earns your trust in the score. Pull back, and you see the context, the entire EGFR kinase domain, its two lobes in rainbow, with our docked molecule nestled in the cleft between them. That cleft is the ATP site, block it, and the receptor can't signal. This is the structural picture behind every targeted cancer drug in this family, and we reproduced it in one short script. So we went fully three-dimensional, a real protein from the PDB, a real binding site, and a docked pose that matched the crystal, all from open-source tools. That's structure-based drug discovery in miniature. Next episode, we scale everything up, virtual screening, scoring a whole library of molecules at once and ranking the best candidates. See you there.